I'm a soil microbiologist, and I've been sort of drawn into this field of taxonomy by the pure need of it and by some lucky circumstances. The need is very much because I work with soil fungal communities, and we keep finding all these unidentified sequences, and we want to be able to identify species across studies and compare and find patterns in the diversity that we have. And the lucky events that enabled us to describe this new class is that in the lab where I did my PhD, we were always encouraged to isolate important ecologically relevant species. We were always trying to isolate from root tips in the field to get cultures to be able to do experiments. And that's how we ended up having this Archeorhizomycetes in culture. So we saw this figure on one of the earlier days. It's a 16 tree which places the Archeorhizomycetes within the Taphrinomucotina. And the resolution is not excellent yet within the Taphrinomucotina, but I think we'll be able to sort that out in the future. We have the genomes now sequenced for these two species that we have in culture. And with that, we hope to be able to resolve the phylogeny within. So I've put in these pictures of the morphology. And they have this typical spaghetti and meatball morphology, as we call it. We have the Swedish isolate, which is the Finlayi. It has a lot of spaghetti. It has some swellings on it, which we think some of them are some type of chlamydospores. Others are just, they look empty when we look in sections. The borealis looks slightly different. It has lots and lots of swellings and the mycela is not really very straight and organized. We also have, in one case, it turned into a yeast in one of our cultures. We haven't been able to reproduce that, but it might be actually switching and be more of a pseudomycelia. So that's something that Hector Urbina, who's a postdoc with me now, he's doing a lot of culturing and trying to sort that out. But before we were able to describe this class, we knew that it was very common. And that was because of work that Chris Schatt had done in 2003. He published this study on soil fungal communities in tundra soils, and where they used large subunit sequences and found that following snow melt, there was like a big unknown groups of fungi that dominated the communities, and they could not be placed. So they went on together with Terry Porter and their collaborators and looked in different ecosystems and kept finding this abundant group of fungi, very high diversity, and always associated with roots, as it seemed. At that time, there were like 13 studies available that had also found it in other systems. And the reason that they could detect it at this high abundance, I should say, is because they were using large subunit sequences. And a lot of studies, or most studies of soil communities, use ITS sequencing, and the reversed ITS4 has two distinct mismatches against this group. So that's why it's sort of been not coming up as very abundant in many soil studies. So the main hypothesis of the work that I'll present here today is we think that Archeorhizomycetes is very abundant and that it is a very diverse class of soil fungi. And the challenges that we faced really is how can we identify species in this group and how can we describe them from environmental sequences. So a lot of the work that I do is looking in GeneBank for sequences and finding other studies that found sequences that are similar to Archeorhizomycetes. And in doing so, I came across the studies that Lee Taylor has done extensive sequencing in Alaska. And among the most common OTUs that he detects are the Archeorhizomycetes, so I contacted him and invited myself to analyze this data together with him. And you see, they really did a lot of sequencing. So they had like 52,000 clones. I don't know if I believe that, but that's what they say. They could detect 1,578 OTUs within their Alaska sampling. And when we pulled out all the sequences that are associated with Archeorhizomycetes, we find that in the different studies they have, it's between 1% and 29% of the sequences. We could detect or find that there's probably 59 OTUs, 53 species within Alaska, and 18 of them we could detect more than once within this data set. What you'll see is, those are the names OTUs, that's when I've seen them more than once. We found the 2 red dots on the screen are the cultured isolates that we have described as species. And we did not detect borealis within Alaska, but we did detect the Finlay. So then the other thing we did with that data was that we analyzed the ecosystems that the samples came from. So they have the 2 main systems. It's the black spruce system, which is green, and the mixed upland, which has the yellow. And we use Unifrac to analyze the branches in the tree to see if they are specifically associated with the ecosystems. And we also do the same for the habitats, organic and mineral soil. And we see strong patterns in the data for, like, some lineages will be associated with the spruce habitat. And we have also the same pattern with the mineral and organic soil. So it's making sense towards the ecology of maybe groups of species. And because they're so abundant too, several of these taxa can be used as indicator species for the ecosystem or for the habitat, which makes it very interesting. So there's other people than Lee Taylor have worked in Alaska, and there were a total of 10 studies that we analyzed for this study. The size of the dots here indicate how many OTUs they find at each site. So it varies from 1 to 25. And both abundance and diversity decreases as we go further north, and there's one more point which is like way up there where there was only 1 OTU detected. So when we described the second species, borealis, last year, we did say the same type of thing and pulled all the data in from that we could find from GeneBank that we think are associated with the Archeorhizomycetes. And we see that borealis is the most commonly detected one, so it's been seen 19 times. We have a total of 101 studies where we see the sequences from this class, and 149 OTUs. And 51 of these have been detected more than once. So we're starting to see the same things, which makes sense, then it feels like these real sequences, these are real things. We only have 2 species described, as I said, but we estimate that there's probably 500 at least out there. I put this map on, which is the distribution of the borealis. So from these 19 places, we use the sequences and their locations to see where they are found. And we didn't find it in Alaska. It looks like it has an Eurasian distribution. All of them were found with the pine forests, and then there was 1 from a spruce, so that's why we thought they were like associated with boreal trees. So with the 51 species that we've seen more than once, I proposed something like this system that we could, for identifying these again when we find them again. We see it's a real sequence, it's a real species, they separate from each other, and we can identify specific regions in the ITS1 and the ITS2 that are specific to these species then. So it could be like a putative species diagnosis. So we have, I don't see this, but so you see the name, then there's the number of times that we've recorded the OTU or the sequence. The first time the sequence was, a sequence from this group was deposited in GeneBank. We take that accession number and then say this is the sequence at this position. So that could be one way of pulling out these putative species. So the other thing that we do with this data is that we plot all of the observations on the map, and the sizes indicate the number of times they've been observed, because I pulled several studies together for the European samples, for instance. And the color is then the ecosystems that they come from. And we can see a strong geographical pattern, but that might be a result of the sampling, because you see there's lots of samples in Europe and lots of samples in North America, and there are lots of gaps in between. And one thing that we want to do to then increase this reliability of this data is 2 things that we're doing. We're collaborating with the Scottish Soil Survey, so they have this beautiful soil survey. They sample all of Scotland in a grid, Then between 2007 and 2010, they went back and sampled 25% of these plots and extracted DNA from them. So we have now amplified the fungal communities from all of these. That data is not finished to analyze yet, but you will hear about it later. But that will be one way to, there's 183 plots or pits, soil pits, and they have all the soil horizons. They have recorded all the chemistry and the land use and the vegetation, the rooting depth. So we'll have all that information and be able to see patterns among the sequences and identify like habitat specificity of the taxa that we see. The other thing that we've done is go out to other places to try to fill the gaps that we have. And this has been done in a semi-unstructured way by when people go to some field sampling, they also bring this hand tool along where you can extract DNA in the soil, from soil samples in the field. And then they can bring the soil DNA extracts back home for analysis. So we have collected samples over the last 2 years, and this is what Hector has been working with a lot, and he sent me the first data yesterday. So this is the percentages are the relative abundance, so just in sequence counts, how many sequences come from the Archeorhizomycetes of all the fungi. And I should say that we use an alternative ITS4 primer. So we have 2 primers there in the mixture, and then we use a degenerated forward primer, trying to work against this bias of the primer specificity. And we use several annealing temperatures and really try to unbias this as much as we could. We still have the problems with extraction, of course, but we work with the amplification. So we find in the Benin samples is the lowest that we see. It's on 0.6% of the sequences, 1% in Puerto Rico and 0.7% in the Tajikistan samples. And then we have the highest abundance in Sweden. So no wonder we started finding it up there. We have a lower in Indiana is the sample in North America with 9%, but we do have a higher diversity there with 39 OTUs, so the second number is an estimate of the number of species. So with this, we're going to start again looking at the patterns among these sequences and see what species do we find globally and what seems to be specific species. And I think we're going to be seeing a lot of new species. Also the Thailand sample that you see had 31 OTUs. So I'm expecting to see new species come up in those samples. So it's a fairly abundant group, and it's very diverse. So really the question we have now, why don't we have more cultures? Because it should be out there, and why don't we have any records of it in other ways? And I think this is probably fungi that only lives as mycelia, so we won't be able to find any structures in the field. At least no one's found any so far. And the cultures we have all come from surface sterilized root tips, and we've done thousands and thousands of root tips over the last years trying to get new cultures out, and really has not been successful at all with that. And so I think it takes a lot of luck to get any of these ones in culture. The other thing there might be is the conditions of culturing. Because maybe we're not using the optimal conditions, so maybe that can be modified, but that's a very extensive work trying to find specific media and promote the growth against other fungi. Recently also we did a little side project on Arabidopsis roots and found, we looked at like surface cleaned roots, so like inside root in the rhizosphere and in the bulk soil. And surprisingly, we found the highest abundance in the rhizosphere soil. So it might be that the ones we find in roots, they just occasionally make it in there, but mostly the rhizosphere is their preferred habitat. So that would be another way to sort of explore more, where can we isolate these? Or we should just go with the sequences. So this is my concluding slide. I think Archeorhizomycetes as a class is most abundant in typical ectomycorrhizal ericoid type of habitats, so along the top of, like decreasing towards the top and towards the equator. Remember not to use the ITS4 primer if you want to see it. And I think the good thing with this, we have all these sequences that we can use to maybe suggest putative species, and the good thing with this group is there's no risk of competing with any existing descriptions because I don't think there are any. The other thing we need to do, because it's really hard to resolve the relation with just the ITS and LSU sequences, I think we have the advantage here that we can get samples that are maybe dominated to 40% by this one species. So if we just screen a lot, we can pick out samples that are good, and we could do like metagenomes and get whole genomes from the environment, or we could do really, like large fragments by PacBio sequencing, and would maybe be able to get environmental sequences into good classifications. Thank you.